We just finished talking about transcription. You now know how the messenger RNA is copied from the genetic code we call DNA.
So what's the next step?
The next part of protein synthesis is translation.
Translation? That sounds like something going to be translated from Spanish to English.
That's because that's exactly what happens. Do you remember how the genetic code in DNA is transcribed to mRNA in the nucleus?
Yes, DNA was transcribed to mRNA with adenine pairing with uracil, thymine pairing with adenine, cytosine pairing with guanine, and guanine pairing with cytosine.
Well, now mRNA moves out of the nucleus through nuclear pores towards the ribosome in the cytoplasm. It is taking the code that is transcribed from DNA to be translated by tRNA.
So the code goes from DNA to mRNA to tRNA?
Exactly. Now the tRNA has to translate the code that mRNA has into specific amino acids.
Amino acids. I remember that amino acids are the common monomers of proteins.
Yes, the tRNA will translate the code into amino acids that will link together to form proteins. Now, the way mRNA is translated by tRNA is pretty similar to transcription. Adenine will pair with uracil, uracil will pair with adenine, cytosine will pair with guanine, and guanine will pair with cytosine.
So A pairs with U and C pairs with G? Got it.
3 nucleotides in a row in the mRNA make up one codon or triplet. Each codon matches an anticodon in tRNA.
How many nucleotides make up each type of codon?
3. At the ribosome, each codon or triplet on mRNA matches one specific anticodon on tRNA. Each tRNA molecule carries an amino acid.
So if I have a DNA code of AAT, CGC, then the mRNA code will be UUA, GCG. What will the tRNA code be?
If mRNA is UUA-GCG, then the tRNA will translate that to AAU-CGC. Now, we take that mRNA code 3 bases at a time to make a codon, and that codon will translate to the amino acid produced.
So the codons are based on the mRNA and would be UUA and GCG.
Right. Now the codon UUA will translate to make the amino acid leucine, while the codon GCG will translate to make the amino acid alanine.
What happens to all the amino acids once they're produced?
The amino acids will bond together using enzymes to make proteins.
I remember this. Amino acids are joined together by peptide bonds using dehydration synthesis. But how does the ribosome know when to start a new protein?
Each mRNA sequence begins with a start codon and ends with a stop codon. There is one start codon, AUG, and 3 possible stop codons, UAA.
This is a review of protein synthesis. Remember, the DNA located in the nucleus opens up and one side of it is transcribed into mRNA. The mRNA then leaves the nucleus and goes out into the cytoplasm where it finds ribosomes. This ribosome is right here, and tRNA comes in with amino acids, and the anticodon right here joins with the codons on the RNA, and this protein is produced, building one amino acid at a time, adding to make a polypeptide. This diagram shows what's happening in translation. Notice the parts: the ribosome and the cytoplasm, the mRNA that just came out of the nucleus after being transcribed. Notice the bases in groups of 3 that are on the mRNA. The tRNA has the anticodons which match the mRNA, joining the amino acids with other amino acids, making a growing polypeptide chain. Here's a close-up look at tRNA. So tRNA, remember, hangs out in the cytoplasm of cells, and the codon of mRNA meets with the anticodon of the tRNA, and the amino acid that's on the end gets linked with other amino acids to form a polypeptide. This is the ribosome with the RNA that's moving through it. And do you remember the 3 bases at a time are called codons? And they match with an anticodon, which is on the tRNA. And the amino acid that's on the end of the tRNA gets added eventually to make a growing protein or polypeptide chain. You're going to have some practice this week on how to read this codon chart. So you take the 3 letters at a time from the mRNA to look to see what amino acid it will join with at the ribosome. So, for example, if you take a G, a C, and an A, what do you end up with? First you start with the left side, so G, C, and A. This is where they all intersect. G, C, A, and you end up with this amino acid called alanine. Here's a different codon chart that helps you get the same result. We're going to find G, C, A and figure out what amino acid comes from that mRNA. We'll start here in the second center of the circle. G, C, we're looking at this quadrant right here. G, C, A, and the amino acid we get is alanine. Each mRNA sequence begins with a start codon and ends with a stop codon. There is one start codon, and it's right here in the chart. It is A, U, G. Every amino acid and protein sequence begins with a start codon and ends with a stop codon. There are 3 stop codons. The first 2 are going to be U, A, and U, A, G. The third one is going to be U, G, A. All those tell the protein to stop being made. So we're going to talk about the overview of this process. The order of the nitrogenous bases is the genetic code. The genetic code is common to all organisms because every living thing contains DNA. So protein synthesis that we just learned about is a process of gene expression. So what is coded in the order of the bases in DNA determines what proteins are made. And proteins determine many characteristics within a cell. So when the gene's code is used to produce a protein, that gene is considered to be expressed.